Cell biology is a fascinating field that encompasses the study of the fundamental unit of life, the cell. Within cells, there are various organelles with unique functions that work together to maintain the overall health and survival of the organism. In this script, we will explore the intricate world of lysosomes and endosomes and their role in cellular processes. Lysosomes are membrane-bound organelles that house a wide array of enzymes responsible for the breakdown of macromolecules. These enzymes are synthesized in the endoplasmic reticulum and then transported to the Golgi apparatus for further processing. Once fully matured, the lysosomal enzymes are packaged into vesicles and transported to the lysosomes. Within the lysosomal membrane, there are various proteins that mediate the transport of macromolecules into and out of the lysosome. These proteins include lysosomal membrane glycoproteins, such as LAMP1 and LAMP2, which are involved in lysosomal biogenesis and fusion events. The lysosomal acid hydrolyses play a pivotal role in the degradation of macromolecules within the lysosome. These enzymes function optimally under acidic conditions, which are created by the proton pumps present in the lysosomal membrane. Some of the key lysosomal acid hydrolyses include proteases, lipases, nucleases, and glycosidases. These enzymes work together to break down proteins, lipids, nucleic acids, and carbohydrates, respectively. Their activity is tightly regulated to prevent damage to cellular components outside of the lysosomes. Lysosomal trafficking involves the movement of lysosomes within the cell to ensure efficient delivery of cargo for degradation. This process relies on the interaction of lysosomes with cytoskeletal elements, such as microtubules and actin filaments, as well as molecular motor proteins. Microtubules provide tracks for the movement of lysosomes by utilizing motor proteins such as dyneins and kinesins. Dyneins transport lysosomes towards the cell center, while kinesins move them towards the cell periphery. This bidirectional movement enables the positioning of lysosomes in specific cellular regions. Lysosomal storage disorders, LSDs, are a group of genetic diseases characterized by the accumulation of undegraded substrates within lysosomes. These disorders can result from defects in lysosomal enzyme activities, lysosomal membrane proteins, lysosomal trafficking components, or lysosomal biogenesis factors. Some well-known examples of LSDs include Gaucher disease, Tay-Sachs disease, and Niemann-Pick disease. These disorders can lead to a wide range of symptoms, including neurological impairment, organ dysfunction, and skeletal abnormalities. Treatment approaches for LSDs often involve enzyme replacement therapy, substrate reduction therapy, or gene therapy. Endosomal recycling pathways are important for sorting and returning cargo molecules to their proper locations in the cell. This process helps maintain cell balance, regulate plasma membrane composition, and recycle components for membrane remodeling. Sorting receptors interact with cargo motifs to guide molecules for recycling via transport vesicles. Endosomal protein sorting complexes play a pivotal role in guiding cargo molecules to their appropriate destinations within the endocytic pathway. These complexes include retromer, Eschert, endosomal sorting complex required for transport, and GGA, Golgi localized, gamma-ER containing, ADP ribosylation factor binding, proteins. Retromer complexes are involved in the retrieval of cargo from early endosomes to the trans-Golgi network or the plasma membrane. Eschert complexes mediate the sorting of ubiquitinated cargo into intraluminal vesicles within multivesicular bodies. GGA proteins are responsible for the sorting of cargo between the Golgi apparatus and endosomes. Endosomes undergo a series of maturation steps to efficiently sort and transport cargo molecules. Early endosomes, formed by the fusion of clathrin-coated vesicles with the plasma membrane, gradually mature into late endosomes, also known as multivesicular bodies. During endosomal maturation, the pH of the endosomal lumen gradually decreases, reaching its lowest point in the lysosomes. This pH gradient is essential for the proper functioning of lysosomal acid hydrolysis and the sorting of cargo into appropriate vesicles for degradation or recycling. Retrograde transport machinery plays a crucial role in the recycling of molecules from endosomes to the Golgi apparatus or other organelles. This retrograde transport is mediated by specialized protein coats, including COPI, coat protein complex I, and retromer. COPI-coated vesicles transport cargo molecules from the Golgi apparatus back to the endoplasmic reticulum, ensuring proper protein localization and maintaining organelle integrity. Retromer complexes, as mentioned earlier, are involved in retrieving cargo from endosomes, allowing its return to the appropriate cellular compartments. Rab-GT pathways are essential for vesicle transport in cells, particularly in lysosomes and endosomes. They regulate membrane formation, transportation, tethering, and fusion. Specific Rab-GT pathways control different steps in these pathways. Activation and localization are regulated by GEFs, gaps, and effector proteins. Lysosome-related organelles, LROs, are specialized compartments that share certain characteristics with lysosomes but have distinct functions in specific cell types. LROs include melanosomes and melanocyte, platelet-dense granules, and cytotoxic T-cell granules. These organelles provide an environment for specialized processes such as pigment synthesis, blood clotting, and immune response. They possess a unique complement of lysosomal enzymes and membrane proteins that enable their specific functions. Endosomal sorting receptors are integral membrane proteins that recognize and sort cargo molecules within the endosomal system. These receptors often possess cytoplasmic sorting motifs that interact with the sorting machinery. One example of an endosomal sorting receptor is the mannose 6-phosphate receptor, which recognizes lysosomal enzymes carrying a mannose 6-phosphate tag. This receptor allows the transport of these enzymes from the Golgi apparatus to late endosomes, where they are sorted into lysosomes. Lysosomes and endosomes are highly interconnected through a network of vesicle trafficking pathways and membrane fusion events. This communication enables the exchange of molecules, cargo, and information between these organelles. Lysosomes can receive cargo from early and late endosomes through vesicular fusion. Additionally, lysosomal membrane proteins involved in lysosomal exocytosis can fuse with the plasma membrane, releasing luminal contents into the extracellular space. The communication between lysosomes and endosomes is crucial for maintaining cellular homeostasis and executing diverse cellular functions. To summarize, lysosomes and endosomes play crucial roles in cellular processes like cargo degradation, recycling, and organelle communication. Their intricate machinery and regulation help maintain macromolecule balance, remove unwanted substances, and support cellular health. Knowledge of these organelles offers opportunities for treating lysosomal storage disorders and related conditions.